Our next speaker, I'm sure, is known to many, friend of many as well. David Altshuler is our next speaker. I had the pleasure of meeting David back in 1997. I'd been a postdoc a couple of years at what was called the Whitehead Institute for Center for Genome Research. Here showed up this guy who was, just a guy coming to do a postdoc. At the time, I guess we were about maybe 30 or so. This is a fellow who ended up really being a leader in everything that we know in genetics in terms of patterns of variation in the human genome, being leader in the SNP Consortium, 1,000 Genomes Project, and at the same time being world leader in terms of identifying and understanding variation in terms of type 2 diabetes. This one friend of mine ended up becoming a founding member of the Broad Institute, which is now one of the leading genetics and genomics institutes in the world. It's not just me that says it. He's also a member of the Institute of Medicine, member of the American Academy of Arts and Sciences, and Barack Obama thinks so as well, since he named him a champion of change. So without further ado, I would like to invite David Altshuler, who is Executive Vice President, Global Research and Chief Scientific Officer at Vertex Pharmaceuticals. And you can understand why I wanted him to come and talk to you, because he has such a unique vision for where we need to be going. Thank you, David.
Well, thank you. I have to tell one story about John, because no, you can sit down. No, it's actually an amazing story. We had a collaboration at the Whitehead MIT Center for Genome Research that was with Bristol Myers Squibb, Affymetrix, and Millennium Pharmaceuticals, called the Functional Genomics Consortium. And it was about looking ahead. Eric Lander, who founded the Broad Institute and was the head of that, he's a very foresightful guy. And so that same year in 1997, he was saying, well, we need to think about what we'll do once we have a genome sequence. And I remember John Ryu and Mark Daly giving a talk about genetic variants they had found that were associated with risk of inflammatory bowel disease. And it was incredibly exciting because this was one of the first couple of times anyone had ever succeeded in finding any genetic variation related to any disease. But also there were complexities. There were actually multiple genes in the interval, and you couldn't quite tell which gene was the cause. And also there was a pattern of genetic variation, these haplotype blocks that Mark Daly described. And I remember somebody, who's probably Eric, saying at the end of this presentation that John and Mark gave to this group of academics and companies and the like, he said, if you look ahead 10 or 15 years, that's what human genetics is going to look like, because the patterns that we had seen of both the insights, because there was a real insight there about the genetic base of inflammatory bowel disease and the challenges, and it pretty much turned out to be true, actually, that so much of what was seen in that pioneering example. And so it's fun to be here with John and with Bartha and Peter and Philippe and others, good friends, all. But I also wanted to say I'm here also because in my new life, I switched a year and a half ago to working at Vertex Pharmaceuticals, where we are developing medicines based on genetic insights about disease. I do also want to say there's a sort of local pride, because we have 4 research sites in the world, and one of them is in Laval, and actually Camille and Joe are sitting right there from Laval, and doing actually very exciting work trying to develop new therapies for genetic diseases. And so I'm hoping that in addition to my getting to talk to you, there's some new relationships that will form between our colleagues, because we're really engaged in this enterprise together. And so my talk is going to have 2 pieces. The first is going to be somewhat of a perspective on the science of human genetics and how it relates to the discovery of new medicines. And then I'm going to specifically focus on the example of cystic fibrosis as done by my colleagues at Vertex. And hopefully between the two, there'll be some lessons about, at least I've taken away, about genetics and healthcare and personalized medicine and all those things. So this is a picture from the White House a year and a half ago when Barack Obama announced his precision medicine initiative. And you can see he's sort of pointing at this double helix. And it begs the question, what exactly is precision medicine, and what is our genetic what do our genetics have to do with the treatments of diseases? Now, there are different definitions of precision medicine. I'm going to give you one in a second that I'm not saying is the right one, because to some extent this is one of those terms that is impossible to tell one group of people how they should think about it when they might have a slightly different definition. But here's how I and we think about it. A precision medicine is a medicine that targets a root cause of a disease shared by some specific group of individuals as defined based on genotype or another basis. Now, the reason it says or another basis is if you say it's genotype, then people will say, but there are other ways to define what groups you're interested in. But the flip side I would point out is if you sort of say based on anything, it just becomes the practice of medicine. Right? Because in fact, as a physician, I'm trained as a physician, of course we don't give all the same treatments to everyone without regard to any information about their pathophysiology. You try and actually understand why the person with shortness of breath in front of you might have congestive heart failure instead of pneumonia, instead of asthma, instead of cystic fibrosis. And so I think that we have to have some limitations on this, otherwise it's simply not useful. But I think that the essence of this, and I don't want to lose the essence, is that the idea that by addressing something that is a root cause of the disease rather than that is symptomatic, there's the potential to modify the course of disease or prevent it or cure it rather than simply ameliorate it for a brief period of time. And this is actually a deep difference, actually, between what some of our therapies do and others not. And so when we think then about, well, how are we going to get these precision medicines? This is one important disconnect I sometimes feel comes in, because I do sometimes feel as in our excitement, and it's justifiable excitement, I think, about all the data we can measure and all the ways we can begin. The Netflix example was given, you know, how Netflix can maybe guess what movie you want to see, that it's as easy as that in medicine, that if you could say, well, you're in this subgroup, then you should take this medicine. That implies the medicine already exists. Right? And that's actually a very hopeful thought, but I think it's a completely incorrect thought. The idea that when a new mechanism is understood about disease, that all of a sudden we'll just pull something off the shelf and that the hard part is actually to figure out who needs it, rather than that the hard part is to say, we didn't know before that CFTR mutations were the cause of cystic fibrosis, and it's going to take 20 or 30 years to develop treatments based on that. But once we know, then we'll know which patients to give them to. And I think that that's just the reality, is that in most cases, if there's anything we've learned about discovery science and human genetics, is that when you discover you newly discover the underlying cause of a disease. When I heard, I wasn't involved in the discoveries, when John and Mark got up and said, here's this IBD5 regions and these interludes, you had to say, well, how is that affecting inflammatory bowel disease? If you wanted to develop a treatment, whether or not, excuse me, it would be genotype specific, you need to go down that long road. And in fact, that road, and I find, having sort of bridged these two worlds now, that there are different parts of the ecosystem that understand different parts of the journey. And I want to sort of put them up here because I think they're all important, and they're actually quite different in their characteristics. So the first is, we have to understand the root causes of human disease. And that is typically and correctly done by academic scientists who do genetics and other types of studies and understand that there's something going on. And that takes somewhere between, you know, a decade and infinite, because, right, there's many diseases we don't yet understand. We can't say how quickly it'll happen, but it's always the case when some new discovery is made that it takes years to understand it well enough to feel like you really have a picture of what's going on. Now, at that point, when you have enough knowledge about the underlying biology of the disease, you can then start to actually discover the therapeutic. And this takes somewhere like, and I put 5 to 10 years, but again, the sort of far end of these things is, you know, undefined, because there's lots of treatments we don't yet have. But this is not just actually what often is talked about, which is maybe I'll have an assay and I'll do some screen of compounds and I'll maybe get a hit. That's actually not the hard part. That's actually the easy part. The hard part is to find one that has the properties you need, not just of potency and efficacy, but also safety and oral availability or the half-life. And there's a million things that compound has to have to actually be a safe and effective therapeutic, the potential to be. And that takes somewhere on the order of, call it 5 or 10 years and tens of millions of dollars to find that. And that's when you start doing clinical trials, is when you have that, then you can actually start doing clinical trials, and that typically also takes 5 or 10 years, and that can cost hundreds of millions of dollars, because you actually, the onus is on you, as it should be, you, the the sponsor who is claiming this, to then demonstrate it's safe and effective. And that's sort of the order in which you go. You start with safe, and you say, can I actually just give this to someone's body without hurting them? And then you have to show some evidence of efficacy that would justify a large and definitive pivotal trial to show it's efficacious. So this is like a long road, right? And again, if it so happens that the precision medicine discovery is for medicine that's already gone through the second and third bullet points, then you're going to have some real low-hanging fruit, because you'll be able to say, newly, you can just take this medicine that already has gone through the years and decades of discovery and apply it, and that occasionally happens. But I think that's not a general solution. For our children, for our grandchildren, to cure the diseases that still cause suffering, we're going to have to go down this road. But I still believe that that first step of understanding the human disease is absolutely foundational. And the reason I believe that's the case is because in this industry, and by the industry I mean healthcare, not just pharmaceutical industry, but that we're all engaged in, there's a vast amount of investment in things that don't work. Now, these slides are about pharmaceutical and biotechnology drug discovery, where it turns out that when you hear about those budgets, they're fairly large budgets spent on R&D, the vast majority of them are spent in late-stage clinical trials, phase 2B, phase 3 trials, because that's actually a very expensive activity. It turns out it's not researchers pipetting for the most part, or studies in animals, or even those early studies of safety. It's largely in the definitive, large-scale randomized trials. It's a very expensive activity. And it turns out that most of them fail. They fail to show adequate safety and efficacy. And in fact, today, most of that failure is due to lack of efficacy. And that's because people have gotten very good at actually identifying early in the process potential toxicities, such that you don't end up in late-stage trial of things that typically fail for safety. They typically fail for lack of efficacy today. And then if you ask, why is that the case? Well, I think in most cases, not all certainly, but in many cases, it's simply because the therapeutic hypothesis being tested doesn't apply to the patients. Okay, in which it's being tested. It could be that in someone else that would be an effective therapy, or it could just be that that therapy is not doing something that would be helpful for anyone in particular. And so it turns out that not only does that gate the productivity of the pharmaceutical industry, but of course the productivity of the pharmaceutical industry in turn gates how good are the medicines that physicians have to offer their patients. How good are the medicines that patients have and families have to ameliorate their disease? So this is not a small matter. Right? And so it says that there's something incredibly important back at that early stage of what is the cause of the disease, to make sure you're going down the right road. Because that road, if I've built any argument, is that that road is long, it's expensive, and it fails a lot of the time. You don't get to the other side, and that's a big cost to society. Not only the true cost of money spent on things that don't work out, but I think even more importantly is the opportunity cost of things we could have done that would have helped somebody that didn't get done because we didn't have the right process. And so the argument I'm making is that, many therapeutic hypotheses, at least in recent decades, have actually come from an understanding of cell and molecular biology, not of people, but actually of cells in a dish or of animal models. And that was because there was tremendously powerful biology that could be done, and more power to it. But that led to a situation in which the first test of the hypothesis in patients was the late-stage clinical trial, because there was no human data, for the most part, prior to the clinical trial that tested whether or not modulating that target would actually help a patient. And many companies actually have looked at and published, actually it's quite interesting, someone asked about pharmaceutical companies and data sharing. Actually, there have been a lot of self-reflective articles written by companies. This is one from AstraZeneca, where the new leadership of AstraZeneca research looked back at what the previous leadership of AstraZeneca research had done. But they looked at what was their success rate, and they were very honest about, first of all, the fact that if you looked, and again, this is a published paper people can read, that in their as the trials were in early, as in preclinical even, before it went into patients or phase 1, most of the failures were for safety. For example, in this study, in their experience, in phase 2B, 88% were for efficacy. And then they showed that if there was human data, human genetic data linking the target to the disease, prior to the onset of the program, their success rate was perhaps twice as high, or about twice as high. And that makes sense in a way if you look at what this enterprise is. So to me, that logic, which is actually not about precision medicine really, it's not really about human genetics, it's not about data sharing and reciprocity of IRBs, it's just about the importance to the enterprise of understanding the underlying basis of human disease. And so we all know, and I'm not going to belabor the point, it's known to the people in the audience and also be the subject of much discussion, that we are truly living through a transformative period, by which I mean not the last 2 years, but the last 30 years, in our understanding of the genetic base of human disease. This plot, which was published actually over about 15 years ago, shows the discovery of genes for single gene, so-called Mendelian disorders, and you can see that there were essentially like a handful in which the underlying molecular basis was known prior to about 1985, 1990. And the reason that this curve took off was because of two things. There was the idea of how you would create a human genetic map that was published by David Botstein and colleagues in 1980, and then there was the publication of the first human genetic map in 1986, I believe it was, which actually, amazingly enough, Mark Daly, his friend and my friend in crime, it was like 19 years old and is like the third to last author on that paper. But that first genetic map was published in 1986, and you see right thereafter, that tool was so generally useful that all of a sudden the positions of genes were identified, and then using the tools of positional cloning, genes and mutations were identified. But as you also can see here, there was almost no change in human complex disorders during this period of time, because it turned out this model of linkage worked only for diseases that had very strong single gene effects. But as we already heard, the Human Genome Project and databases of genetic variation, and basically just tools that allowed you to do systematic genetic epidemiology, comparing genetic variation in disease, then made it possible for common diseases to identify as well the much more complex and myriad genetic risk factors for each disease. But today there are thousands of those identified as well that connect to common diseases. And this is just a call out to the colleagues with which I spent 15 years developing these resources. And I always have to remember, because I'm going to touch on CFTR for the second half of my talk, that the inspiration for that genetic epidemiology approach and the HapMap project and all that was not actually Francis Collins and Eric Lander suggesting it one day in a bar, which sometimes pundits would like to suggest. It was actually that the mapping of cystic fibrosis actually was enabled by haplotype mapping. And people may remember this paper from 25 years ago, which was the paper describing the cloning of the CFTR gene as responsible for CF. But they don't always remember what the red and orange and yellow and blue and green bars are behind the little boy with CF. And what those are the haplotypes at the CFTR locus. And the red haplotype is the haplotype carrying delta 508, which I'll talk about later in my talk, that actually was, and when they basically had positional cloning and got within millions of bases of the gene, they found that very surprisingly, the majority of kids with cystic fibrosis had the same haplotype at that locus, and only a very small number of people in the general population did, and they recognized this was a founder haplotype, and that's how they mapped the gene. And this was the inspiration for doing haplotype mapping more generally in common diseases, which ended up being productive. And so one sort of statistic, which is, again, remarkable as we try and wade through the thicket of hype and promise in this area of personalized and precision medicine, is that if you go back 15 years when we started mapping genetic variation, and you ask the question, if I were to take Peter Goodhand's genome, and I were to sequence it, and I were to look up what fraction of the genetic variation in his genome is already in a database somewhere where you could look up what the consequences in terms of disease association were, the answer would be like 0, because there was no public database of genetic variation, let alone a database of how that variation tracked with traits. That's only 15 years ago. Today, 99% of the genetic variation in any person's genome on the planet is in that database. There's something like 60 or 70 million genetic variants in the public database based on the 1,000 Genomes Project, and there's over 100,000, probably now I've lost track, 200,000, I don't know how many people have had their exomes sequenced. And all that data actually is in a public database where you can discover the identity of the variation, and meaning that you know that there is a variant with that frequency in that population. And so if you sequence someone's genome, it turns out over 99% of the variation in that genome is already sort of tabulated. That is a really interesting and important sea change to go from none to most. But what value we derive from it, we're still working out. So the argument that I've come to, and I'm going to illustrate this with two examples, is that really what we need is to be able to triangulate on the right assays, the right laboratory assays and targets for drug discovery, and this is now on the where do the new precision medicines come from question, using this knowledge of phenotypes and genotypes. And the reason it's in a triangle is there's sort of these three parts of it if you look here. We have to start with the human population. So we're not starting again with the mouse population or the population of cell lines in a dish. We're starting with people. And we have to understand the relationship between genotype and phenotype. And that is something that, all the work that Bartha was talking about will be discussed at this conference is continuing to drive forward larger and larger data sets, understanding which genetic variants track with which phenotypes. And it's important that it not just be one phenotype at a time, because part of what's valuable, and again I'll illustrate this with an example, is not only knowing what you thought you wanted to know, which is this gene might be involved in this disease, but also if you're going to make a pharmaceutical agent that's going to modulate that, you'd like to know what might happen that I wasn't looking for, right? And so if you only have one phenotype at a time, you won't figure that out. You need to be able to look across phenotypes, and since genetic variation around the world is varied and heterogeneous, sometimes you can learn a lesson in one population that can inform others around the world. So you need to, that's what really the genetic mapping problem is really about, that left side of that diagram. But then you have to be able to go from that knowledge that this gene and this variation plays a role in disease to some sort of laboratory experiment that you could use to drive therapeutic discovery, because you can't just think about it and say, okay, that gene plays a role and magically a pharmaceutical or a medicine is discovered. You need laboratory tools. And that's a tricky business, because it's not always the case that a knockout is the same as the physiology you're interested in. So sometimes people say, well, it's great, we'll just take that and we'll make a mouse knockout and we'll study it. But of course, there's no reason to think, first of all, the mouse will necessarily recapitulate what you're doing. And it's not clear that actually complete loss of function is the same as whatever the genotype, phenotype that you had is. And so you need to very, very carefully find that assay. But I actually think that's a big, not say the hard part, because remember, the next 10 or 20 years is very hard too. But if you don't have that right, if you have the wrong assay, then again, you're not going to be able to succeed. And if you have the right assay, well, there's still a tremendous amount to do. It is very enabling to be able to say, now we really believe that this assay does faithfully recapitulate what we need to do in the patient to modify their physiology and help them, and then you can try many different approaches to try and achieve that. And just one article, if you want to read more about this, in 2013, Robert Pledge, Ed Skolnick, and I wrote this article called Validating Therapeutic Targets Through Human Genetics that analogizes the dose-response curve that you're going to see both for safety and for safety and efficacy in your clinical trials, and just tries to analogize that to the perturbation in phenotype for sort of desirable and undesirable consequences in response to a genetic perturbation, and say if you can actually see that when there are successful medicines and where there is genetic variation, you often see that analogy. And in particular, if you don't see any relationship between your target and what you were hoping to do, that also might guide you. And I'm going to now illustrate these 2 points before I turn to CF with the story of HDL, LDL, and coronary artery disease. So everybody knows in this room, I'm sure, that LDL is associated with heart disease, coronary artery disease, such that higher levels of LDL are associated with higher risk of heart disease, and higher levels of HDL are associated with lower rates of heart disease. And this has led to them being called the bad cholesterol and the good cholesterol. And I was trained as a physician, and I actually became a diabetologist, board certified, all that stuff. And we were trained that you wanted to lower the LDL, and you wanted to raise the HDL. And how many people in this room have ever had it suggested to them by a physician that raising your, or a newspaper, that raising your HDL would actually benefit you in terms of your coronary artery disease risk? Yes, people raise their hands, because that was actually the practice of medicine and probably still is. It is absolutely clear that lowering LDL does lead to reduced risk of coronary artery disease. I don't want to leave any misapprehension on that point. And that's actually because both through human genetics, epidemiology, and critically, LDL lowering therapies through multiple targets, you do see that relationship hold. But what about HDL? So this asks the question, of course, how do you test the hypothesis that HDL modification would be causally involved in coronary artery disease? And there's really only 2 approaches that I think can get at causality. One is a randomized trial, an experiment of man, or woman, where basically you randomize people to an exposure, and then you evaluate what's the outcome. The other approach is to use genetics in what's been called Mendelian randomization, where there's a genetic variation that affects the variable you're interested in this case HDL, and then you ask the question, and you design your study right so that your groups are matched except for the flip of the coin in meiosis as to whether they got that allele or not. And then you ask, is it the case that inheriting the allele that modifies HDL actually modifies the outcome, in this case, coronary artery disease? And so two postdocs in my lab, Ben Voigt and Seik Kathreson. Ben on the left is now a faculty member at UPenn. Seik is now the head of the Center for Human Genetic Research at Mass General and a faculty member there. But they were both postdocs in my lab years ago, and they said, we've done genome-wide association studies, and others have, and we found genetic variants that affect LDL and HDL and coronary artery disease. We should be able to look at this triangulation. And so they published this paper, and the long and the short of it, it was published a number of years ago, so I'm just going to go through a couple of high points, is they first recapitulated what had been shown earlier, which was that genetic variants that affected LDL all were associated with altered rates of heart disease in the direction and magnitude of effect that you expected, and there were actually, I think, like a dozen different genetic variants in that study. So it wasn't just one gene and one genetic variant, all 12 went in the right direction, right magnitude of effect with heart disease. And then they asked, what about HDL? And the answer was that individually and collectively, there was no consistent relationship between HDL alteration and heart disease. And this slide, which is just sort of one of the examples, this is a functional variant in the gene endothelial lipase. So it's functional, meaning that it's not one of these non-coding GWAS SNPs. It's actually in the protein coding region. It had been shown to affect the function of endothelial lipase. Endothelial lipase had a known role in HDL metabolism. And when we first looked at this and showed that this functional variant, which in other figures in the paper does show the predicted effect on HDL, had no effect on heart disease, we sent it to a leading journal of medicine published in Boston, Massachusetts, and they said, not enough samples, not enough. So we went back and got more and more, and I think actually in this there's 117,000 people with and without heart attack for whom that this was tested so that we could get the confidence intervals very narrow, and you can see that little diamond at the bottom overall, which is 95,000 controls and 20,000 cases of coronary artery disease, functional variant. You can see like there's not a big spread on either side because it was down to like very tight confidence intervals. And actually, they still didn't want to publish it, so it was published somewhere else. But actually, the point of this paper was that there was no consistent relationship. Now, in the years subsequent to this publication, actually, a year before this publication or two, one clinical trial had failed quite spectacularly, torcetrapib for Pfizer, a medicine, a CETP inhibitor that doubled HDL cholesterol with an expected quite profound effect on coronary artery disease risk, and it actually had the opposite effect. So we published this paper, and there were genetic variants in CETP and other genes. But nonetheless, since then, I think there have been 4 other large clinical trials published that have shown large effects on HDL of a variety of different niacin and CETP inhibitors, not one of which has actually showed an effect on coronary artery disease in the desired direction. So in addition, there is this interesting out-of-left-field human genetics observation that variants in CETP, cholesterol ester transport protein, is the target of multiple of these agents that raise HDL and have not yet succeeded in any of the published data in lowering coronary artery disease risk, although there are ongoing studies. This paper found, surprisingly, it was a study not of heart attack, but of age-related macular degeneration, and it actually found that genetic variants that were associated with raising HDL, which is what you would want to do for coronary artery disease, was associated with an unexpected 70% increased risk of age-related macular degeneration, something you might not have thought to look for and you might not have been looking for. But based on this, it is a p-value of 10 to minus 22nd and a 70% increased risk of effect on age-related macular degeneration. So that could be something to look for if you were modulating CETP. Now, that's on HDL. And so that's just an example where, independent of discovering a new target, that actually is, I think, useful information. If you're contemplating spending hundreds of millions or billions of dollars, as society does, and it's not just pharmaceutical companies, there's still lots of funding from nonprofit funders and researchers, and by the way, there probably is some way to modulate HDL or some aspect of that physiology that will actually benefit patients. The fact that the serum level, as measured in these studies, does not track doesn't mean there's no way to do it. You can never prove a negative. It's just about what's been found. But what about LDL? Are there new targets? And again, as well known, so I'm going to do this quite quickly. Here's where human genetics, again, was quite useful. There was a family-based linkage study of a very rare family with high cholesterol. They found this gene PCSK9, about which nothing was known, and it was figured out these were gain-of-function mutations. Then Helen Hobbs and Jonathan Cohen showed that there were also common loss-of-function mutations in African-derived and European populations that were polymorphic, frequencies of about 0.5 to 1%, and because they were common, those variants in PCSK9 that were loss of function could be used in genetic epidemiology studies exactly of the sort that I'm describing here. And so it was figured out they not only lowered LDL, but they lowered heart attack risk. And then an individual was found, 2 individuals, who had homozygous loss of function and no immunoreactive PCSK9 in their blood. So this told you that lowering it a lot was not, at least in the context of genetics, associated with any adverse events. And so that combination of a new target, an effect on a biomarker, LDL, an effect on an outcome, and complete loss of function not being deleterious is a very compelling package to look at. And so that's a case where even though statins are terrific, they actually have certainly not eradicated heart disease, which is still in fact the leading cause of death in the world, and blood pressure lowering and smoking cessation and all the other things have been done. And so now there are, as you know, 2 approved medicines that came directly from this observation. So the final point, and then I'm going to switch to CF, is that those examples might not strike you as precision medicine because it's not like subsetting people. It might not seem like, and then, although actually you'll note that HMG-CoA reductase inhibitors were initially developed for familial hypercholesterolemia. And if you look at the initial approvals for PCSK9 drugs, they're actually, and I'm not going to comment on exactly what they say, but you can read the labels, and they're for specific subsets of patients. But what happened with HMG-CoA reductase inhibitors, statins, and may happen with other medicines, is that once there's a medicine that's in some context safe and effective, then further clinical research is done, clinical trials, and you can potentially expand the label. And with statins, initially, those were thought to be only for people with secondary prevention and very high rates of cholesterol, but by the time 20 years had gone by, in fact, it was shown not only that at any LDL level there was a reduction in risk of heart disease, but Paul Ritker's Jupiter study looked at people who did not have any elevation in LDL cholesterol, but instead have high CRP levels, and they showed in that study that that really seemingly different group of people without the so-called LDL sort of a contribution to heart disease, they benefited from a statin too, suggesting it's more of a quantitative rheostat rather than it's like a defined boundary of this group versus that group. And so I think it's important to realize even if something is developed in a subset where there's the clearest potential for the right benefit risk, it doesn't mean that's the only place it will ever be useful. All right, so I'm now going to switch gears from that sort of historical and conceptual view to then share with you the work done by my colleagues at Vertex for CF, which is a really terrific example of precision medicine. And I'd like to say that I can't take any credit for this work because it was all done before I got there, but I'm certainly proud to work with this team. And Fred Van Goor, whose picture is there, he gave me these slides, and he's led this program together with others for 15 or more years. So I think people know cystic fibrosis is a disease. About 75,000 people around the world have CF. And the disease was initially, the name cystic fibrosis was because people would get dilatation of ducts, cysts, and scarring, fibrosis. And it was a disease where people died very young in life. I'm talking about 25, 30 years ago, and they would die young in life. And mothers had noticed that when they kissed the babies, and fathers, that they tasted salty. And in fact, the diagnosis was done by a sweat test, where you would actually measure the chloride in the sweat of a baby, and if it was very high, then that was told you it was cystic fibrosis. And then, as I showed you earlier, it was identified that this disease is caused by mutations in the gene CFTR that is involved in chloride transport. And there were many valuable treatments that were developed for this disease, but none actually aimed at reversing the underlying CFTR defect at the time that I'll pick up this story. And just to give you a sense of the timeline, the gene was discovered in '89, and within a few years it was shown that it was a chloride transporter. And there was tremendous work done by lots of people, I'm going to describe some of it, to understand the genotype-phenotype correlation and also the mechanisms of those mutations. But even after work had gone on for 5 years at Vertex, leading to the synthesis of the compound known as Kalydeco or ivacaftor, which is approved for treatment of patients with specific mutations and age groups for CF. It actually took 2 years then before the first clinical trial starts, and then 6 years to approval. And then, as I'm going to tell you more about this story, and then 3 years after that, a second medicine, which is actually a combination of ivacaftor and another medicine called lumacaftor for people with a different genotype. And so I'm just going to take you through this story, which is a rather long road, but a very satisfying one. So the academic community had worked out that this pathophysiology of CF, of the mutations leading to defects in the CFTR protein, then defects in chloride transport, this leads to thickening of the mucus, clogging of ducts, infection, and then you end up with the clinical manifestations of lung disease, pancreatic insufficiency, failure to thrive, infertility. It all makes sense, if you will, based on this biology. But nonetheless, there were a lot of challenges to this story because there were many different mutations. And although that's a challenge, it also turned out to be very informative. Those different mutations have different mechanisms and severity of effect on the protein. It's a multi-organ disease, so you can't just study necessarily one cell type and expect to take care of all the problems. And of course, there's many other things that affect the severity. Even someone who has the same, two people with the same mutation, there are many other things, not just genetic and also environment, infection, how they care for themselves, et cetera. So it's actually, for a so-called simple genetic disorder, it's still surprisingly to some complicated. But the genotype-phenotype correlation, the different mutations that were found, actually were incredibly informative. This plot, which shows the study of decades of work understanding different genotypes, and then their cellular consequences and also the clinical consequences, shows on the x-axis for different mutations the amount of CFTR activity that remained. On the y-axis, this measures sweat chloride, which again was used diagnostically years ago and can be measured in patients. And then what you can see is that people who have carrier levels of CF, if you're heterozygous, this is autosomal recessive, if you're heterozygous, there's no phenotype. Carriers, essentially, they have perhaps slight increases in sweat chloride, but they actually are phenotypically, from a clinical point of view, they're fine. And then you can see that there are mild mutations that have CF-related diseases and more severe mutations with perhaps later onset, and then the most severe mutations with the most severe phenotype. So this told you that the assays you would want to use to actually study treating this should recapitulate this behavior. Now remember, I was saying a knockout is not the same. You could imagine if you only had 2 data points here, there might be many features of cells in which you took out CFTR, and they might mislead you. But here you have a quantitative relationship, a dose response. And in addition, I don't have a slide that shows this, different mutations were found to have different mechanisms of action. Oh, I do have a slide. And in fact, what those were, you could understand that previous slide, which was the relationship of CFTR activity to clinical consequences, as a product of the amount of protein on the surface and then its ability to conduct chloride. And so the consequences of these two things, the amount and the activity, really led to the phenotype. And so this really is an exquisite example of being able to triangulate between these different features of the human population, the genes and variants, the cellular assay, to then figure out what you needed to do in therapeutic discovery. The 2 other key things here were the use of human bronchial epithelial cells. So this was not done, the workhorse of this project, or to keep it focused, was actually patients would donate a lung either because they were having lung transplant or because they died. And scientists at Vertex and other places would collect the cells, the human bronchial epithelial cells, and then culture them with different genotypes. This is before you could work with stem cells and before you could do gene editing. This was the only way that you could have human cells that actually were differentiated and had specific genotypes. But that meant that the studies were actually in a highly relevant context. And they were also, because this was an ion channel, they were tools to measure the gating of the chloride flux from the protein, and also you could measure the location of the protein. So again, it's a little bit different than sort of having some generic approach to how you're going to study every protein coming out of genetics. It's very tailored to the specific physiology. And so terms were coined in the field long before we were doing it, or before we had done these things, that a protein that corrected that led to an increase in the quantity of CFTR on the surface, in particular in delta F508, which is a processing mutation would be called correctors, and one that would increase the gating in the would be called potentiators. So here's an example of a particular genotype, G551D. This is a defect in channel gating. So this protein makes it to the cell surface, but actually doesn't open. And you can see here in the patch clamping studies, open and closed state of normal CFTR and how it's very seldom open in G551D. But then after application of ivacaftor, this is in vitro, you can actually see the channel opens and you see increased chloride transport. And again, this was actually many years between identifying a compound that could do this, then actually find one that could be safe and effective and have pharmaceutical properties was a different challenge. And then this was then studied in those clinical trials that went on from, remember, synthesized in 2004, first studies in human beings 2006, and approved in 2012, basically going through phase 1, phase 2, and eventually pivotal study to show the effect on both short-term and long-term benefits to patients, and as well as characterize the safety profile and tolerability of this. And I would refer you as to published data and the label if you're interested in specifics about for whom this is approved and its balance of safety and efficacy. In delta F508 homozygotes, which do not have a gating mutation but have a processing mutation, both the Western blot in this case and also immunostaining could be used to show the cytoplasmic CFTR in the upper right where the mutation didn't have any correction of that gating of that processing defect, and then after treatment with lumacaftor, a fraction of the protein then makes it to the cell surface, but it turns out actually it has a gating defect. If you take delta F508 and you treat with this agent and get it to the cell surface, there's a gating defect, but it can be treated with ivacaftor. And so the medicine, Orkambi, is actually a combination of lumacaftor, which for patients who are delta F508 homozygotes and currently only approved for 12 and above, age 12 and above, you have something that gets the protein, improves its processing, gets it to the cell surface, and then also ivacaftor, which affects the gating. So this is really kind of a remarkable story because of many things. It's a remarkable story because of the collaboration of academic scientists and medical scientists and patients, because we should not underestimate the importance of the Cystic Fibrosis Foundation and the patients and families who worked with them in defining how to care for patients with the disease, putting together care centers, supporting the basic science that understood this, and even working together with companies like ourselves to initiate these drug discovery programs, but then also actually over 15 years of work to actually not only implement assays at high throughput, but actually discover compounds and then go through all the process of figuring out how to make them pharmaceutically, have what are called pharmaceutical properties consistent with treating patients chronically, and also to demonstrate that in different studies. And we're really nowhere near done with this. The amazing thing about even a single story like the CF story is that even though this has been going on now from 1989, gene discovery, in 2012, Kalydeco, in 2015, Orkambi, in our minds at Vertex, and I think very much in the minds of the patients and families struggling with this disease, we have a lot more work to do. Only a fraction of the people with CF actually are currently approved for one of these medicines. 24,000 out of the 75,000 are now approved for one of our CFTR modulating therapies. But actually, even for those medicines, there's lots of clinical trials being done to look at other genotypes and other age groups, just to see if those medicines can be shown to be safe and effective so that people can have access. But then in addition, there are patient populations for whom it's likely these medicines will not actually be adequate. And we have a pipeline, we have medicine in phase 3, in phase 2, 2 in phase 1, with different properties for different genotypes or patient populations. And it'll no doubt be many years till we can reach our goal for just this one disease, which is to have therapies that can bring people to carrier levels even at early in life, and to show that you have to start later in life and then work your way backwards to show safety and efficacy for children, and that would actually apply to all the different genotypes. And so you can see it going all the way back now to gene therapy and gene editing kind of approaches we're looking at. And so this is, on the one hand, you could say, wow, that's a lot of a lot of time and effort to go into this disease. On the other hand, there's 75,000 people with life-threatening disease, and this is a pretty remarkable story. And there's lots of other diseases like that. And as I hope to sort of convey in comparing the LDL-HDL story and this rare disease story, is that it's kind of not helpful to actually, in my mind, to separate rare and common diseases if they're fundamentally different. There are very important differences, but the logic, which says that we're trying to understand the relationship of patients, phenotypes, and genes and variants to train assays that then will actually succeed in the clinic, I think is the same, although the challenges in terms of implementing that strategy are certainly very different. Now, before I close, and then if there's time, I'll take questions, I just want to sort of say a couple of things. First of all, there are a lot of limits to this idea of using human genetics for target identification and validation. It would be inappropriate not to mention them. So the first is, only a subset of potential targets that would be valuable will happen to carry mutations in the human population, and that's because it might have something to do with it just never occurred, or it might have to be that in evolution, such mutations, for reasons we might not understand, are deleterious, and it might also be that the fact that you have to go through development with a mutation, embryonic development, puts a different filter on things than if you were going to treat later in life with a medicine. So only a subset of potentially great targets will have such properties, but on the other hand, the ones that don't have this information or the information can't be obtained through some other means have a different risk profile, because you have to sort of have a leap of faith that absent such information, whether it's genetics or pharmacology or something else that tells you about the pathway in patients, you have a leap of faith to go from your model to the clinical trials. Common variants are typically not knockouts and provide only partial perturbations of gene function, and actually CF is an example of that. Right? There are knockout null mutations of CF, but actually it's very informative that there are mutations that, although more severe than your typical common variant, nonetheless were partial loss of function or specific mechanistic defects. They can be such mutations can be highly informative, but it's important to realize that they don't tell you the maximal effect that you will get. They only tell you something about the relationship between genotype and phenotype. It's sometimes been said, and I think it's just not correct, that if a genetic variant has a modest effect on something, then ipso facto it's not of interest, let's say, for therapeutic discovery. Well, I think if it's a knockout and you're going to make an inhibitor and it's a complete knockout, then that's a pretty good proxy for what your inhibitor would do. But if your genetic variant is actually a 5% change or a 10% change in function, and you actually can go further than that with a therapeutic agent, then you can learn more. And I think that the PCSK9 story is actually an interesting example of that, where the genetic variants that are found actually do not have as big effects, most of them, as you can achieve with the therapeutic agent. And of course, you then there is some risk as you go beyond what you've seen before of what will happen, but nonetheless, it's important to realize that quantitative relationship. There's this important point about variants being present from conception on and all through development, and a treatment that you're going to give typically would be later in life. And there's two elements of that. One is, as I've said before, you may not be able to obtain the evidence you want because there's some other role in development, you can't see it. But it's also that the timing of when you would apply such a therapy is hard to know. It sounds really good, the idea, and it is really good, the idea that we could identify patients based on genotype before they got sick and then intervene to prevent the disease. But if you think about practical clinical development, you have to think about how early do you have to start, and how long do you have to follow people, and what are you going to follow, because there has to be some practical clinical trial. You can't, otherwise you can never demonstrate the value. And actually that's something really, because the genomics community often does say things like, we can start beforehand and prevent the disease, and I think the implication is often that we don't have to prove those things work, it'll be all lifestyle or something, and we'll just say, but that's first of all not reality. And we are obliged, and we should be obliged, to actually test our hypotheses in well-performed clinical trials. So, for example, if you think about the thought experiment, it's just a thought experiment, that somebody were to develop a pill, and it's just a thought experiment, a pill that in BRCA mutation carriers could prevent breast cancer, okay? Let's just imagine you could develop such a thing. Think about how you would do clinical development. It would actually be very difficult because women, first of all, have surgery now that is effective. And so would people not have the surgery and then go into the trial? They'd have to know whether or not they did that. There's a risk there. So do I think it's impossible? No, I actually think it's something that we should really think about, because it'd be much better to have a pill that you could take that would, or something that rather than having to have oophorectomy and mastectomy prophylactically. But it's challenging to think about it. And what it comes down to is you need things to measure, biomarkers or some sort of early tumor markers or something you could measure that would allow you to have enough early warning. And we work on other disease, Huntington's disease. And again, you think about if you had something that you could show would prevent Huntington's disease, but it didn't reverse it once it had occurred, it had to be done prophylactically, then again, you have to have a biomarker or something you could follow, or else you can't do the clinical trial. And actually, in this regard, I think the FDA is actually very forward-looking. They've actually done a number of things in recent years. A number of policies have come out and pathways that are about, I think, the wise application of, especially for rare diseases with very strong relationships of this sort, where the people really do get the disease, how do you put in place a different profile of how you develop that medicine? Because you can't do all the same things you could do, and on the other hand, you have the advantage of knowing these people will progress. And finally, the last bullet point here, knowledge of genetic association is hypothesis generating, clinical trials are needed. In our space, that's not a problem, because no one would let us do this without clinical trials, but we think that's great because we want to perform. We did a 1,000-person placebo-controlled randomized trial for Orkambi in CF, a disease with 75,000 people. 1,000 of them participated in that trial. So that's the kinds of things we do. But I think in some cases you do see people wanting to leap from the genetic association to the use of medicines. We see this in cancer more than you see it in inherited disease, where people say, well, there's this relationship and it works in a dish. And I leave it to everyone in the room to think about, how much evidence should we need to actually show that the predictor that's being used to personalize care actually leads to better outcomes. I'm personally more comfortable with there being clinical trial data to support it. Although once you have a strong clinical trial base, then as you think about where you go from there, it's different to have, let's say using the cancer example, to have a number of mutations or a number of genes where you have done those studies and then going to other mutations rather than never having any such data. So I'll just close with these two slides. My first day of medical school, I was handed a book written by Francis Weld Peabody, who was a great Harvard doctor who died young. And in this book, there was this quote, "One of the essential qualities of the clinician is interest in humanity. For the secret of the care of the patient is in caring for the patient." And I thought, as a 21-year-old, I thought, that's the dumbest, who would not be interested in the patient? But I discovered over the course of now, it's been 25 or 30 years actually, that I've now been in this business, that actually a lot of times the things we're doing for patients don't actually come from clear thinking about patients. This whole talk is about thinking about patients first. Sometimes it comes out of scientific theories and models of disease, and then the patient is lost. So I've sort of modified this, one of the essential qualities for the development of new medicines is interest in human biology. For the secret in prevention treating disease for the patient is understanding the underlying biology of the patient. And I think those are the stakes for the things we're talking about in this conference, but we can't be naive about how hard it will be and how long it will take to see benefit. The question I always ask is, if we don't do these things, how long will it be till there's a treatment for something like CF? So thank you very much.
We definitely have some time for some questions, so please, once again, use the microphone in the center.
Thank you.
Thank you so much for this beautiful talk. My name is Yves Tremblay. I'm coming from Laval. There is another variable that you have to think about. That's the difference between men and women. And that's also the gender question. So gradually all of those things will come in and will probably complicate the analysis of data. But it does not apply to the CF. But people have to think about that.
Yeah, no, I think you're absolutely right. I think that what you're saying, which again I think is best done in patient data, and which is why we need the kinds of information resources that are, Bartha was talking about, is that you can't just assume that it's the same in men and women. You can't just assume it's the same in different groups around the world. You need to actually have data to look. And so even if it's the case that the elegant scientific theory that came from a theory of cell biology or from perhaps a genetic study or something else that then went into mouse models and has been extrapolated. We can't just assume that that will apply everywhere. But the good news is that the kinds of information resources that are being developed should make it possible to ask questions like the one you're asking.
Yeah, that's true. And one of the best examples of that is about the asthma. How the clinical trials have been done in South Africa and try to export this to Europe. And you see that there is so many problems with that.
Yeah, I mean, the other thing that's really interesting, we had a wonderful talk at Vertex the other day in our seminar series by someone about pain clinical trials. So as people probably know, there are these, there's a terrible problem with opioid addiction, and we clearly need new pain medicines. There haven't been new pain medicines in many years, and there are multiple approaches, but one is based on human genetics, the NaV1.7 mutations that have been found that, as people may know, loss of function of both copies of NaV1.7 is associated with complete insensitivity to pain. And so multiple companies, including our own, have developed agents that modulate that target. And we had someone come in who actually he was in clinical development. He was an expert in pain trials. And he was taking us through all the ways. It wasn't about the science of which target and which compound and everything. It was just all the different ways that how you run the trial can actually affect the outcome. One example is you don't want people in your trial, let's say, who don't have much pain, because there's no way to improve on not very much. So many trials historically, this wasn't about a Vertex trial, this was just a story he told, would say you should only get into the trial if you have above on a scale of 1 to 10, a 5 or something like that. Well, that's upwardly biased, right? So you ask people, do they have a 5? And anyone who says 4 doesn't get in, anyone who says 6 gets in. But even if there was some noise around that, let alone bias, it'll tend to regress towards the mean. And so you end up with the quote-unquote placebo effect not being something fancy. It's just that if you ask people to jump over a little bar to get into your trial, the almost always going to fall right after they get over the bar. And so there's so many things that we have to do to get this business right. And of personalizing medicine, if we're really going to do from genetic science to discovery of new medicines to proving their value and providing access to them, how we do the clinical trials will matter, in whom we do them. And there's no substitute for rigor. I mean, I think that you have to be rigorous. You have to remember questions like, are men the same as women? Are people from this group or this severity of disease going to be the same as the people from whom I base this?
Sexes, but every person is gender.
Yes. Any other questions? Yes.
I'll give people another minute or two to get unshy to ask you a question.
Yes.
Where do you see, I mean, now that you've only been there for a year and a half or so, how do you see the interactions going forward between academia, pharma, and all the other players that's going to be essential if we really want to try and achieve success in this area?
So this is kind of ironic, actually, because maybe it's ironic, but because I was someone, as John knows, not everyone knows, but certainly very much inclined towards collaborative science, and for example, that global alliance that I was involved in starting, has companies and all that. So you might think that that was my bias, and now working in a company, I might somehow have a different view. But actually, I have the same view, but it's even more informed. And the reason I say that, meaning that it is important for us all to interact, is because, look at the CF story. The knowledge of the genotype, phenotype, biochemistry of that came from academia. But actually, the 15 years of drug discovery and knowing how to make a small molecule that had all the properties needed, and then doing clinical trials, something that only happens in companies. And actually, the regular exchange between scientists at Vertex and in that CF community, not just in the science where that started, but also in the clinical trials, and then what happens when you actually get on the market and people are using it, is an incredibly virtuous cycle. And I think that we find, and again, that's one of the reasons why I was excited to get this invitation and to have Camille and Joe join me here today and have people get to know each other, is in Montreal, there's a microcosm for what needs to happen. We can't possibly do everything within this research site that goes on in Montreal. We're going to do a tiny fraction, but there is expertise in what we do and the other companies in this region do that doesn't exist in academia, or just the ability to really focus on a project for years and invest what you need to invest to make a medicine. and not only for the discovery of it, but then the testing of it. So I really believe that it's even more important that academia and companies work together. And I think that part of the reason to give a talk like this and to try and lay out the different pieces is that I find that the different parties often only think about the piece they do, right? So if you're, so again, speaking as a lifelong academic, I kind of, academics often know about the academics discovering the biology, and they often know actually about the sort of later stage clinical trials and getting on the market, if they're academic physicians at least, because they're involved, or physicians, they're involved in that. But actually the decade of work in between, you don't see. And so often people will talk as if doing a screen and getting a hit is like discovering a drug. But in fact, there's about a decade of work between doing the screen and getting a hit and actually having a drug. And so at the same time, people in companies who may not be involved in the early discovery or in the prescribing of the medicine or even the clinical trials, who are in research, they may not understand those pieces. And so there's just no substitute for actually having these conversations, because we only succeed, and this is the scary part, but it's why it's amazing it happens at all. We only succeed if you get all the way from the beginning of this road to the end. Like there's no close in horseshoes or whatever. Like it doesn't help if someone publishes a paper, finds a gene, has an assay, gets a hit, and then, you can't get it to be safe enough, or, you do the wrong clinical trial, or, if you can't get all the way to the end, no patient benefits. So we have to talk to each other, because different parties are actually doing different parts of this process, and we only have any value really to society other than knowledge if we get all the way to the end successfully.
Thanks. That was a great talk. There's a tendency to emphasize disease-causing variation, even in the context of GWAS, where there could be some protective alleles. But there was this neat paper that came out recently where they identified a small number of individuals with what looked like very high penetrance disease, kind of Mendelian disease genotypes. And as far as you could tell from the way the database was constructed, they probably didn't have the phenotype. And certainly those of us that use EXACT find lots and lots of loss of function mutations. So I wondered if you could comment on the potential utility of real high penetrance protective variation and how we can find it and how we can use it.
Yeah, no, I think it's first of all a very important idea that genetic modifiers can be used to understand, ways of helping someone who, despite the presence of a highly penetrant mutation. I think that actually the history of this, it's interesting, that paper, the Resilience Project or whatever, had excellent ability to tell their story, but I think actually there were earlier work by lots of people that had done it actually even more seriously, because that was really an example of a direct-to-individual response. It was great, and getting these large data sets, but actually there are large modifier screens that have been published for Huntington's, for BRCA1, for BRCA2, for other diseases where people have actually, for example, the BRCA community had 10,000 people in a database, and actually I collaborated on one of these studies. I was not the prime mover, but a large GWAS study, and then later other studies done for BRCA1 modifiers of penetrance, BRCA2. The Huntington's community has done large modifier scans. So I think the idea of doing that work is very well motivated, because it does tell you in the presence of a highly penetrant disease mutation, what can ameliorate it. But I actually tend to think the way to do that is the way that these consortia have done it, where you really meticulously make sure that the people in your study have an actual disease mutation, and you followed them over time and really know what their phenotype is. Because the trouble of doing it is like people self-reporting is you're going to end up probably with some genotyping errors. They may not really have a high penetrance mutation, or the phenotype data may not be right. You have to be very careful, I think, to do that thing well if you're going to sort of look for the exception to the rule. But I think that as an approach, it's a very strong approach, because sometimes the disease mutation itself may be too hard, or at a minimum we have one shot on goal, the Huntington's gene. If there's a modifier of Huntington's that can ameliorate that, that gives you at best, at least a second shot on goal, and perhaps something more practical. So I think the point is very well taken. It's a very important thing to do. And at some level, PCSK9, or examples like that we published. Philippe discovered a gene a number of years ago, and the first GWAS study of type 2 diabetes was done, collaboration of Philippe and other colleagues, and they found this gene SLC30A8 through a GWAS study, and then a large collaborative group that I was a part of showed that rare loss of function mutations in that gene reduced glucose levels and risk of diabetes despite obesity. and older age. And so even though that's not a highly penetrant mutation, it's sort of environmental and age as a risk factor. It's the same idea, and I think you can walk in either direction. You can walk from identifying a gene through GWAS and then find other variants. There's another couple of nice papers, actually, Philippe, who's here, may talk about tomorrow, of rare and common variants in the same gene as another way to get at this.
Great. With that, I'd like to thank David for a wonderful presentation.
Okay, thank you very much.
I have a couple of announcements, but please, thank you very, very much.